This PPT deals with the study of microorganisms in air. The content of this study include microorganisms in air, that is outdoor microflora, indoor microflora, sources of the microorganism in air, and factors affecting air microflora, particularly airborne diseases, fungal diseases, and viral diseases. Here this slide shows about the introductions of microorganism in air, that is aeromicrobiology is the study of airborne microorganisms, and air contains significant number of microorganisms, and it also acts as a medium for the transmission of microorganisms. Airborne transmission is one of the important modes of transmission of infectious diseases, and the composition of air includes nitrogen in the percentage of 78%, oxygen 21%, argon 0.9%, carbon dioxide 0.03%, and hydrogen 0.01%. And microflora of air. The microflora of air can be studied under two headings, that is outdoor and indoor microflora. Let us discuss about outdoor microflora. The air in the atmosphere which is found outside the building is referred to as outside air. It is larger in volume than the indoor air. The dominant flora of outside air is fungi, which includes deuteromycetes and basidiomyces. The bacterial genera commonly found in air are Bacillus clostridium, Sarcina lutea, Micrococcus luteus, and Corynebacterium. The air flora over the sea includes Achromobacter, Bacillus, Corynebacterium, Flavobacterium, Micrococcus, Sarcina, and Staphylococcus. The number of microorganisms may vary from place to place. This is mainly due to the difference in human population densities in the world. And next one will be the indoor air microflora. The air found inside the building is referred to as indoor air. The common fungal genera in indoor air are Penicillium and Aspergillus, and the bacteria are Staphylococcus, Bacillus, and Clostridium perfringens. In case of the occupant being infected, other pathogen bacteria or viruses of the respective diseases may also be present along with the previous microorganism which is present already in the body. And sources of the microorganisms in air. Since air does not contain enough moisture and nutrients to support microbial growth, it is not a natural environment. The second thing would be the air does not have any indigenous microflora, and soil microbes enter the air when disturbed by wind and human activities like digging and plowing. Aquatic microflora also enters in air by forming water droplets or aerosols, and splashing of water by wind action or tidal action may also produce droplets or aerosols. Air currents may also bring the microorganism from plant or animal surfaces into air. For example, spores of Puccinia graminis travel over 1,000 kilometers. The main source of airborne microorganisms is human beings. The commensal and pathogenic flora of upper respiratory tract and mouth are constantly discharged into air by the activities like coughing, sneezing, talking, and laughing. Then the microorganisms are discharged out in three different forms. They are droplets, droplet nuclei, and infectious dust. So this is the main sources of microorganisms where humans can get easily affected by the bacteria present in the dust, nuclei or droplets. In droplet, we'll see one by one. The droplets are usually formed by sneezing, coughing, or talking and consist of saliva and mucus, as we all know it already. So the droplets may contain hundreds of microorganisms, including pathogens of respiratory tract origin. The size of the droplet determines the time period during which they remain suspended in air. Most droplets are relatively large, about 10 micron or more. When inhaled, these droplets are trapped on the surfaces of the respiratory tract and cause diseases. If it is pathogenic organisms, it is severe. Then droplet nuclei. In warm and dry atmosphere, small droplets evaporate rapidly and become droplet nuclei. Thus, the residue of the solid material after drying up of a droplet is known as droplet nuclei. These are small, 1 to 4 micron in length. They remain suspended in air for hours or days, traveling long distances. And viability of the droplet nuclei depends upon some factors. They are the atmospheric conditions like humidity, sunlight, and temperature, then size of the particles during the organisms, and the degree of susceptibility or resistance of microbes to the new physical environment. And the droplet nuclei may act as more potential agents of infectious diseases than droplets. Then factors affecting air microflora. The number of intrinsic factors and environmental factors influence the kinds and distributions of microbes in air. The intrinsic factor will be it includes the nature and physical state of microbes. Spores are relatively more abundant than the vegetative cells. So fungal spores are also abundant in air. The size of the microorganisms is another factor. So generally smaller or easily liberated into air and remain there for a long time. The state of the suspension plays an important role in the settling of microorganisms in air. In the free state can settle out slowly because of the weight, whereas the microorganism embedded in the dust particles settle out rapidly and they remain airborne for a short period. Droplets which are discharged into the air by coughing or sneezing or remain suspended in air for short period, when their size decreases by evaporation, they remain for a long period in air. Viruses need 17 to 25 percentage. Then air current is another important factor. If the velocity is high, the particles settle down very rapidly, but gentle air keep them in suspension for long period. And high altitudes are characterized by severe conditions like desiccation, ultraviolet radiations, and low temperature. Only resistant forms can survive. Then airborne diseases. The number of diseases are transmitted through air. Most of the respiratory tract infections are acquired by inhaling the air containing the pathogens, and bacterial disease will be brucellosis, which is a causative organism for brucellosis. Sources will be aerosols from infected swine. Symptoms will be aches and pains of muscles and joints, prolonged undulated fever, headache, and chillness. It affects veterinarian, butchers, slaughterhouse workers, etc. Then second one will be meningococcal meningitis. It is a causative organism will be Neisseria meningitis. Source will be droplet containing meningococcus, and symptoms will be nasal secretion, sore throat, headache, fever, pain in the neck and back, loss of mental alertness. Then pulmonary anthrax. The bacteria responsible for this is Bacillus anthracis. Painful swelling, nausea, coughing with blood, painful swelling will be the symptoms, and the source will be dust contaminated with infected animals, wool of infected animals. And fourth one will be diseases caused by Streptococcus pyogenes. So streptococcal pharyngitis known as sore throat. Symptoms include fever, enlargement of lymph node of neck, bleeding from reddish throat, and scarlet fever, more or less similar to pharyngitis. In addition, characterized by skin rashes, rheumatic fever, which is characterized by inflammations and degenerations of heart valves, and erysipelas. It is a skin infection causing lesions on face and legs. Then puerperal fever, it acquired at childbirth and affected breast. Then fungal diseases, cryptococcus. It is a causative organism will be Cryptococcus neoformans, and symptoms include asymptomatic, sometimes mild pneumonitis, and sources will be inhalations of soil particles containing the virus. That is fungal. Then next one will be blastomycosis. The causative organism will be blastomyces dermatitis, and the symptoms include suppurative and granulomatous lesions in any part of the body, and the sources will be the inhalations of soil particle containing fungus. And X virus will be the coccidioidomycosis. It is inapparent, sometimes fatal. Causative organism will be coccidiosis immitis, and symptoms will be fever, rheumatism. Sources will be inhalation of dust containing arthrospores of fungi. The next one will be histoplasmosis. Causative organism will be histoplasma capsulatum. Symptoms will be asymptomatic, affect reticular endothelial system, and the source will be rotting and soil. Then viral diseases will be the common cold, which is caused by the rhinoviruses. Symptoms will be running nose. Sources will be droplets from nose and throat discharges. Likewise for influenza, arthromycovirus, Symptoms will be nasal discharge, headache, muscle pain, sore throat, general weakness, etc. And next one will be measles. Causative agents will be morbillivirus. Symptoms will be fever, cough, cold, and red blotches, skin rashes will appear. And sources will be the respiratory tract secretions in the form of droplets. And mumps. It is caused by mumps virus. Symptoms will be painful swelling of parotid glands and salivary glands. And source will be droplets containing infected saliva. Then adenoviral disease. It is a causative agents will be adenoviruses and symptoms will be acute respiratory disease and pneumonia. Then significance of air microflora. The microflora in air is very few in number and plays a significant role. So this is due to the fact that the air in it direct contact with almost all animate and inanimate things. In human health, air acts as a medium for the transmission of infectious agents to human beings. Outdoor air does not contain disease causes pathogens in the significant number to cause any infections. In case of indoor air, the chance for the spread of infectious diseases is more, especially in areas where people gather in large numbers, for example in theaters, schools, etc. In hospitals, air within the hospitals may act as a reservoir of pathogenic microorganisms, and infections acquired during the hospitalizations are called nosocomial infections, and the pathogens are also called nosocomial pathogens. The common microorganisms associated with the hospital infections are Haemophilus influenza, Streptococcus pneumonia, Streptococcus aureus, Pseudomonas aeruginosa, and members of the Enterobacteriaceae and respiratory viruses. And there are two main routes of transmissions of nosocomial pathogens. One is contact and air. So that's all about this microorganism. Thank you.